Hi everyone, let us quickly revise electron transport system and oxidative phosphorylation from the chapter respiration in plants within 9 or 10 minutes. Let us understand first the definition. Electron transport system is the pathway through which the electrons passes from one carrier to the another carrier is called electron transport system. The electron carriers are situated within the inner membrane of mitochondria. Now, let us briefly understand what are the components of mitochondria or structures present within the mitochondria. Mitochondria, it consists of the inner membrane, which is having an infoldings called cristae, outer membrane, and the intermembrane space present between outer membrane and inner membrane, and inside the inner membrane is the matrix. Matrix is the site for Krebs cycle or citric acid cycle. When we take a small portion and elaborate it, the inner membrane of mitochondria consisting of various complexes which contain electron transporters. The complexes are complex 1, complex 3, complex 2, complex 4 and complex 5. Let us now understand what are the electron carriers present in each of the complex. Complex 1 consists of it is associated with NADH dehydrogenase enzyme, and it contains electron transporters, namely FMN, flavin mononucleotide, and a complex FES. And complex 3, that is after complex 1 is present directly complex 3, it's not complex 2. After complex 3 is complex 2. Complex 3 is otherwise called as cytochrome B7 because let us now understand why it is called so. Complex 3 is containing electron transporters cytochrome B, FES and cytochrome C1. Hence it is otherwise called cytochrome B7 complex. Then complex 2, it consists of FAD, flavin adenine dinucleotide, and FES are the electron transporters. Complex 4 is otherwise called cytochrome C oxidase. It consisting of two copper ions. They are copper A and copper B. Along with that, it contains electron transporters cytochrome A and cytochrome A3. Here is copper A, cytochrome A3 and copper B. And lastly, complex 5. It's otherwise called as F0 F1 particle. Later we shall understand what is the function of F0 and F1. And before going to the details, we should know one more thing. Between complex 1 and complex 3, there is a free electron transporter that is called ubiquinone, and another molecule of ubiquinone is also present between complex 3 and complex 2. And complex 3 is associated with mobile electron transporter that is called cytochrome C. Now, at complex 1, NADH, which is produced during Krebs cycle within the matrix of mitochondria, will undergo oxidation. We know oxidation is removal of H+. And also it releases electrons. Those electrons are transported through transporters of complex 1 and are carried to ubiquinone. Ubiquinone, using those electrons, will undergo reduction and again it will undergo oxidation. As a result, electrons are given to cytochrome B and the protons are carried into intermembrane space. While transporting the electrons in complex 1 also, the protons are carried into intermembrane space. Now, at complex 2, during conversion of succinate to fumarate during Krebs cycle, there is production of FADH2, and that FADH2 will undergo oxidation at complex 2, and as a result of oxidation, the electrons are given to carriers of complex 2. And those electrons are given to ubiquinone and ubiquinone will undergo reduction, and as a result the electrons are given to cytochrome B, which means complex 3 will receive electrons both from complex 1 and complex 2. Now from complex 2, electrons are given to mobile electron transporter, that is cytochrome C, that will carry the electrons to complex 4, the electron transporters. Now at complex 4, the finally electrons are accepted by a oxygen. So here is the role of oxygen. During oxidation or oxidation of food, the role of oxygen is at complex 4, and it will undergo reduction to form water, and as a result there is a pumping of protons from matrix to the intermembrane space. Now we have observed that between matrix and intermembrane space there is a creation of difference in the concentration of H+. That is, proton gradient is created between intermembrane space and the matrix of mitochondria. Now higher concentration of H+ is seen in intermembrane space compared to the matrix. As a result, protons are carried from intermembrane space into the matrix of mitochondria through F0 F1 particle. F0 particle acts as a proton pump or channel for transport of protons from intermembrane space into the matrix of mitochondria. And F1 has a activity of ATP synthase. Whenever there is a movement of protons from the F0 particle, there is a synthesis of ATP in F1. For every 4 protons moving into the matrix, there is synthesis of 1 ATP, and that is called phosphorylation.